thank you very much. I'll try to tell a little bit of the simulations that we've been carrying out in our group. Some of the results are newer than others, but I'll try to cover two different subjects that we have been studying. Some of my postdocs will present talks later, and they will go deeper into some of these problems. So just to give a flavor, I'll try to show some of the results. I'm not going to be very detailed about it, but just to give you an overall view of the research that we have been carrying out. As you know, the state of São Paulo is a very important, Brazil is a very important producer of sugarcane, especially in the state of São Paulo. For nearly 40 years, we have an industry established, very mature industry established here, that converts, or it's for the ethanol and sugar production out of the battery is gone, out of sugarcane. So we plant sugarcane, extract the sugar, the juices, and we go through fermentation and we produce ethanol. So half of our cars, actually half of our cars run nowadays on ethanol, but somewhere between 70 and 80% of our cars produced today are by fuels. So we can run either on gas or ethanol. So ethanol is an important industry here in the state of São Paulo. However, if you take the whole biomass of ethanol, 1/3 is sucrose solution that you go and you go do ethanol fermentation. But 2/3 of this matter, of this biomass, is bagasse and leaves. And currently, these are usually burned in the sugar mills to produce energy. So the idea is that if you are able to deconstruct cell walls, let's say by some chemical treatment or by enzymatic digestion, you can increase the production of ethanol and other biofuels. If you take the concept of a biorefinery, where you can take the building blocks and build more expensive or more valuable compounds, you can at least in terms of ethanol, you can increase some 40% of our biofuel production without no competition, no land increase, no plantation area increase. So this is very important for a country that should preserve its wonderful biodiversity. So you don't want to increase sugarcane plantation to go into the Amazon and drive cattle ranch into the Amazon. So you can actually, instead of growing plantation, maybe you can help to deconstruct cell walls from decompose sugarcane bagasse and convert into biofuels. So the plant cell wall itself is a very complicated business. It's a polymer. There are cellulose fibers tightly packed. These are surrounded by hemicellulose and interconnected by hemicellulose and lignin. So this makes cell walls very tough, okay, and very difficult to digest by enzymes. And that's why decomposing matter in the woods take very long time. So these enzymes secreted by fungi and by enzymes, it's very long. So usually you need to do some sort of a pre-treatment, either let's say using ionic liquids that are able to do this, to dissolve some of these fibers, or some chemical treatment, or some hot vapor explosion. So you have to somehow get rid of this, the lignin, and expose the cellulose matrix inside. So once the cellulose is there, enzymes can actually go on and attack. So the amount of cellulose, it's the most abundant biopolymer on the surface of Earth. So it's a lot of biomaterials. So there's another important aspect too, where in Europe, the cost of the biomass is considerable fraction of the total production, because it's trees, but here it's already there, because there is a first generation ethanol producing ethanol. So So the idea is that our work is that we work in a very close collaborations with structural biologists or molecular enzymologists. So they extract proteins and they study these systems here, and we actually help them into understanding what is going on. So the enzymes that are involved in this process, it does not just one enzyme that decompose biomass, it's a series of enzymes. I'm here providing a very overall view of what they are. These are glycosyl hydrolases. They usually break the glycosidic bonds, right? And there is some a class of proteins called the cellobiohydrolases. They are exoglucanases. They usually attack crystalline cellulose by the ends, by the extremities. So they bind to the cellulose extremities and they cleave processively from the outside. There are endoglucanases, which usually bind into non-crystalline areas of the cellulose fibers or places where they have defects. Either the action of these two enzymes produces oligomers. Some of these oligomers are dimers of sugar units called cellobiomes. This cellobiose is further degraded into glucose by another enzyme called beta-glucosidases. So they find this oligosaccharide, especially the cellobiose, and cut them into two monomers. Okay? So these enzymes here, usually they have a catalytic core, so they are modular proteins, they have a catalytic core, and they have a cellulose binding module. This cellulose binding module is the module that actually binds to cellulose, so it keeps the catalytic core domain close to its substrate. It's like sitting the kid on a table so that it's close to its food. Okay. So this the cellulose binding module resembles Jim's tryptophan cage. It has 3, or sometimes 2, or sometimes 3 tryptophans that are very hydrophobic, and they look like a paddle. It's a flat bed, and it binds to cellulose. Okay. So it's very similar in nature, at least. Okay, so I hate this. Okay, so the multiscale approach to this is to first understand the plant or the cell walls themselves. How is cellulose assembled? How is hemicellulose bound to cellulose? What is the role of lignin? So there is a lot of action trying to understand the biomass itself. And then you have the enzymes. This is the binding mode, the CBM, or the cellulose binding mode, the carbohydrate binding mode. There are many different types of CBMs. People try to design better CBMs so that you can have a better binding. So, and then you have to understand how this thing binds to the cellulose, how the catalytic core domain lifts up a chain from the matrix, and once this is in there, how the reaction proceeds. So this is a catalytic triad. So in this area here, this region here, you have quantum mechanics. So here you have hundreds of nanometers. Then you have atomistic scale, and then you need quantum mechanics here. Okay? So this is the, let's say, the holistic approach that one would like to understand. In terms of the cell wall structure, as I mentioned before, these are complex systems. You have a fiber of cellulose. You have hemicellulose that covers these fibrils, and then you have lignin. So the access to cellulose by the enzymes is very inhibited if this whole thing is covered by hemicellulose and so on and so forth. So to break this up, it requires usually very expensive thermochemical pretreatments. So it actually, it's an expensive part of the process. And what we'd like to understand a little bit more from the basic aspect is what are the thermodynamic forces that hold these fibrils together and hold this cell wall very cohesive. So we would like to understand the interactions between cellulose microfibers and between hemicellulose and lignin. So for that we use different approaches. Rodrigo is going to talk about some of these approaches that we have. This is not actually molecular dynamics simulation, it's a different approach. But one of the things that we first did was to actually construct cellulose fibers. How do you construct cellulose fibers? So there are many isoforms. They have a crystalline structure resolved. There are some controversies about what is the exact structure because there are different allomorphs, polymorphs. Allomorphs or polymorphs? The biologist is correct. So the structures are very different in terms of cellulose. They're similar but different. So the idea is, how do you actually construct a fiber? So Tiago is a former student. Now he's a faculty at ITA, actually, the Institute of Aeronautics Institute of Technology. So we can construct very long fibers. These are not cartoon. This is actually representation, all atoms. So we can construct, for instance, this is a very, one of the models for elementary fiber in primary cell wall that people believe it is. It's a 36 chain. It depends also on the type of plant that you have. If it's angiosperm or gymnosperm or whatever, it's a grass. Right? But you have this very long fibers, 36 chains in a layer. And these are actually in the cell wall, in the plant cell wall, synthesized to make a bundle of these fibers. And so you have a 7 bundle here, and this is surrounded by hemicellulose. So the first thing that we want to do is can we construct the layers? Can we construct different types of cellulose nanocrystals? So we can construct actually any type of cellulose nanocrystal, like squares, flat beds, things with exposed hydrophilic areas. So this is a very versatile program called Cellulose Builder. It's free. You can actually download it. We are actually now implementing it as a web service, so that you don't have to download the program. Just say what you want there in the website, and it gives you the atomic coordinates for the whatever fiber you want to produce. So this is one thing. So with that, one of the studies that we did was let's take one of these types of fibers, and we want to understand how xylem, it's a type of hemicellulose, how xylem is absorbed, physically absorbed, on the hydrophilic and on the hydrophobic surfaces of cellulose. There were experiments by our collaborator and friend, Paul Webb from Cambridge. He was very much interested in carrying out this sort of experiment. He's a plant biologist. And he found evidence that if you have decorated, so these yellow things here are decorations, are side chains on a xylem chain. If you have decorations, you would only see evenly spaced decorations, but not oddly spaced. Like decoration here, jump 2, have a decoration. But if you jump 3, you would not have you would have a decoration, but it would not bind to cellulose. So the idea was to understand how. So we did that. So we created these fibers, we decorated differently, and we simulate this in solution, and then we simulate this on top of cellulose. And what we find is precisely, but it's, although again it's no thermodynamics real, we are not computing free energy or anything, but we did find that if you have xylem on the surface, the twisting symmetry, it's a twofold symmetry. So if you have an oddly spaced decoration, it will clash on the surface. Only if you have an even instance, because when it's in solution, it takes a threefold screw, the cellulose chain. But on cellulose, it's a twofold screw. So if you have a oddly spaced, you clash. So this is agrees or at least corroborates quite nicely what they were seeing in their plant studies. Okay. So the other aspect we wanted to investigate is to, as I mentioned before, what are the thermodynamic forces on primary cell walls? So we are not talking about interaction between one cellulose and the other cellulose. We are talking about generalized forces. We want to understand the free energy cost of taking let's say, a cellulose fiber apart. So this is a much harder question to answer than just doing simulations and looking at the interactions. So the idea here is that the decorations on the hemicellulose, it's a xylem chain with decorations. This is what hemicellulose is. You can change this many different types of side chains of decorations, and that provides you different types of hemicellulose. And people has actually devised some strategies to genetically modify the organisms, the plants, so that you have plants that produce softer or more amenable to digestion cell walls, but without affecting plant growth. So what you have, you will be able to still have a plant that grows and produces, but the cell walls a little bit more easier to digest enzymatically. So this is one interest overall. So we wanted to understand, so these are supposedly two cellulose fibrils. We have a bath or a whole bath of cellulose side chains. We just put here the side chains, and we wanted to compute the free energy profile for separating these fibers. So we want to see what is the effect of this surrounding media on the binding force between two fibrils. So this is work that Rodrigo did, and he's going to explain a little bit more about that. So here are some of the results that I will let Rodrigo explain to you. So this is some very nice, interesting results. But we can also compute the average density of the solvent of the different types of cellulose, of hemicellulose around the fibrils. So he can actually understand. These are not just cartoons, these are actual population densities that you take out of this calculation, so that you can say, well, this area of hemicellulose is mostly bound to this and this and that. So Rodrigo will be able to tell a little bit about that. Another part of this work is now, it's a continuation, is to understand now what are the interactions now. So now we want to look at secondary plant cell walls. So we want to understand what are the interactions of lignin. Lignin is a very complicated biopolymer, lots of phenolic rings. These phenolic rings has different epoxy groups. Yeah, I don't know the name. Yeah. Okay. So the different types of these units here makes lignin or the plant, the cell wall, harder or softer, depends on the composition of this type of. So trying to genetically manipulating lignin is also another aspect of genetically manipulating the plants that produce lignin, so that the plant can grow and yet be able to be digested more easily for pre-treatment. So the idea is to understand how lignin and hemicellulose interact with itself. Again, thermodynamic forces between different types of lignin and different types of hemicellulose. So that was also what Rodrigo did. And I think it's one of the aspects. Okay. So what we are looking for, we have not done yet, it's one of our proposals, is to have a more complete view of the plant cell wall. So we have layers of cellulose covered with hemicellulose. Have another fibril here covered with hemicellulose. Maybe there is one layer through here to there. And in this space here, we distribute lignin monomers and try to understand this on a broader scale. Of course, this is very difficult if you want to do it atomistically. So we need to go to a larger scale, maybe of course. Now, what about the enzymes themselves? I'm just going to give you a couple of examples there. We've done several studies on this. But we took one of these enzymes, for instance, and this is called the cellobiohydrolase 1. It's from this microorganism called Trichoderma rhizii. This is a fungus, filamentous fungus, that produces a lot of enzymes. If you take the commercial enzymes from Novozymes, people say this is just Trichoderma rhizii juice. They grow the fungus and they mush it together and they produce the cocktail enzyme. It's a very, expresses a lot of enzymes for the construction of plant cell walls. So there is one interesting problem with in this enzymes is the following. So here's the catalytic core. Here's the cleft where the polysaccharide binds to. And here's the catalytic area. And this is the reaction product. It's a dimer. It's a cellobiose unit. There are 2 sugar units in there. So this is the reaction product. It's known experimentally that as the reaction, the hydrolysis, catalytic hydrolysis proceeds, the product of the reaction inhibits enzyme activity. So people try to understand what are the molecular bases. Why the product inhibits enzyme activity. So if you look at the structure shows the X-ray structure shows that the product of the reaction sits on an open cleft. So you think, well, why cellulose just simply leaves the cleft and make room for another polysaccharide to bind, and then you proceed with your reaction. So Rodrigo started looking at this problem a few years ago, and carefully examining the simulations, he noticed that although the crystal structure showed an open cleft like this, as the simulation proceeds, you don't see the backbone of the loop closing like that, but you see the side chains. One of the side chains, actually a tryptophan residue, it actually binds to a neighboring, an arginine, I believe, yes, to an arginine, on the other side, and that traps cellobiose inside. Okay? So very often you see these transitions, so you see the very strong binding between this tryptophan, it's a tryptophan 381, binds to an arginine here, and closes the loop. So you have actually a transition between closed and open conformation, and when it's closed, cellobiose is not able to leave the cleft. So Rodrigo was very excited. We were very excited. Oh, this is wonderful. Let's try to do the thermodynamics. Let's try to understand the binding reality. But actually, then, as we started doing this, people in NRL lab have found very similar results using a different methodology. They actually went into this region here, and they mutated every region in this area and found that the culprit for actually cellulose binding is tryptophan 381. So this suggests that if you mutate this to something else, you might be able to produce an enzyme that is less inhibited by its product. Okay? So this is now another work that we carry out with our experimentalist friends. They have solved this wonderful structure. This is a laminarinase structure. This is the binding cleft. This structure is very nice. It's from a hyperthermophilic microorganism. This microorganism is able to sustain high temperatures, very high temperatures. The enzymatic activity is also very high at high temperature. So it's able to survive up to some 90-something degrees without degrading. And it also has activity at very high temperatures, 80 or some more than 80 degrees Celsius. It still keeps its activity, its best activity actually, optimum temperature. So the idea was to understand why is this enzyme stable under such high temperatures, and why is it still active? So the idea was to, this is just a parenthesis actually. So if you try to understand one of the driving forces that keep the protein folded, right, is that when it's stretched in solution, the protein has hydrophobic and hydrophilic, hydrophobic and hydrophobic, no, this is hydrophilic, I'm sorry, hydrophilic and hydrophobic residues, but when it's folded, usually the hydrophobic clumped in the center and the hydrophilic on the outside. That's a very naive picture, but that's how we like to portray it, so that the hydrophobic core is in there. If you have something that disrupts this hydrophobic core, then you lose protein stability because water goes in there. So we decide as well that maybe we should look at different types of enzymes, similar enzymes. One which is our enzyme, very hyperthermophilic. The other one not so much, maybe a thermophilic, and the other one a mesophilic. Mesophilic means that it will behave as a regular protein at 40 degrees, 80 degrees, 60 degrees, it's already gone. Okay. So we took proteins similar. This was work that was carried out by Erika, by Tiago, by Leandro, and Rodrigo, and other students. So the idea was that, well, let's look at the protein, different types of protein. Our protein, this is the one that we have, it's hyperthermophilic. This one is just thermophilic. And this one is structurally analogous, but it's just a mesophilic. And what we saw is that we performed these simulations under different temperatures. And what we saw first was that the proteins that are highly thermophilic, they have one water molecule inside the hydrophobic core. That's a structural water, it sits there. If you increase the temperature, it will be there, but no other water molecule will come in. In this protein here, which is a mesophilic, you have the water there, but as you increase the temperature, water immediately starts to migrating from the outside into the interior of the protein. So you have something like a contamination of the hydrophobic core by water molecules. And this stabilizes the structure. And we know why these water molecules go in there. This protein has, in the hydrophobic residues that make up the hydrophobic core of the protein, it's actually, let me see it here. It's somewhere between here, these two sets of beta sheets. There are some salt bridges in there, in this one here. There are some salt bridges that permeates the hydrophobic core. So it's like you have a hydrophobic core with something very hydrophilic in there. So the hydrophilic portions allows water to go in. It's like having a very tight wall here, but one very easy breaking point. So where they had the hydrophilic salt bridge, water will go there, and then that's an invading point for water goes in. So that explains, at least in an ad hoc sense, why is this protein so strongly resistant to thermal denaturation, whereas a mesophilic is not. So we also wanted to understand its thermophilicity. That is why one of these enzymes loses activity when you increase the temperature, whereas the other ones do not. So what the students did was to look at the conformation where the binding of the carbohydrate is there. So what we found is that there are, as you do the dynamics, you don't see this in the structure because the structure is static. There are open and closing motions. And you have obstructed binding cleft, you have open binding cleft, so this would allow the substrate to bind. This one also would also allow the substrate to bind, but this one here would not allow the substrate to bind. So the three enzymes have these three swaps, these three conformations. But as you increase the temperature, okay, as you increase the temperature, this is the predominant conformation in the mesophilic. So as you increase the temperature, this obstructed channel conformation is the predominant conformation in the mesophilic. So this means exactly that. As you increase the temperature, the mesophilic will not function anymore because its binding cleft is closed. This does not happen to the other ones. The thermophilic and the hyperthermophilic, open and tube-like states predominate. Okay? All right, so much for this. I want to spend like 10 minutes, very fast, talking about a different types of proteins that we have been working. Actually, this is work that was actually initiated in my lab by Leandro Martinez when he was still in diapers, I guess, many years ago, right, Leandro? And this was the work that first got me involved in trying to understand and working on proteins, which is the field of nuclear receptors. So now this has nothing to do with biomass and all that anymore, but it has to do with human health. So nuclear receptors are proteins that participate very active in the regulation of gene transcription. Certain genes, they regulate its activity in the body. Usually genes that are associated with cell differentiation, development, cell homeostasis, so this has to do with metabolism in the grown body. So it has importance in diabetes, cancer, inflammatory processes, and metabolism. So what this hormone or this nuclear receptor do, they act in the cell nuclei, and they bind to the DNA, and their regulation is made by means of the binding of hormones. When hormones binds to these proteins, they are active. When the hormones are not bound to these proteins, they are not active. So then when they bind to the protein, the transcription of genes takes place. So this process here is not just cartoon. This process people can actually take a picture of this. So this is an actual picture of it's an electron micrograph where you see the DNA here, and you see there's a whole machinery here. But these are the RNA growing outside of that. So this is actually microRNAs being synthesized out of the DNAs by a machinery, a protein enzymatic machinery, very complex over there. But you can actually see the nucleic acids. Okay. So the superfamily of this nuclear receptor, many families, right? You have the thyroid hormone receptors, you have vitamin A receptors, vitamin D receptors, steroids, estrogen receptors, progesterone, testosterone, glucocorticoids, fatty acids. So you see that these nuclear receptors, they are very important for commercial development of pharmaceuticals, but they also have broad range of application in different types. So there are some like 48 families in this superfamily, and 28 of them, at least in the humans, are known to have some specific binders, right? So as you can see, these are those. I know that this room is mostly composed of older men, and there are a few older women here, but see, lack of vitamin D, lack of estrogen, hormone reposition, this is very common to us, right? Okay, so in our families at our age. So the thyroid, for instance, is a very important one. It concentrates the blood cholesterol. It has implications in lipid metabolism. It also affects the heart cardiac activity. So there are especially two subtypes of this hormone. One is highly expressed in the liver. One is highly expressed in the liver. So there's this one here. And the other one is predominantly expressed in some types of muscles and the heart. So if you take hormone to activate the thyroid hormone, it's going to have a beneficial effect of increasing your metabolism in your liver, but it will also ruin your cardiac activity because it's going to activate both. So the pharmaceutical interest is to develop a ligand that is selective to the beta isoform and not the alpha isoform. So it activates in your liver and not in your heart. Okay? All right. So I already mentioned this. When I can go here, but well, yeah, I can go. So in terms of structure, these are Leandro's slides.
Masterpiece is slides.
What?
They're my masterpiece.
Your masterpiece is slides. Yes, it's a keep on. It's a renewable resources. This is the global structure of this protein. It has a ligand binding domain, it has a DNA binding domain, and has an N-terminal domain here that nobody actually knows what it does. But these modular proteins, here is usually where we work, the ligand binding domain. So this binds to DNA, but this binds to the ligand. Okay, so we want to understand that sort of thing. So there are many different types. They all have the same type of structure. It's like a sandwich of alpha helixes. They don't look similar here, but they are similar. You see a couple of long alpha helix in the back, and then you have other ones in the middle, and then ones on the top. All of them has this structure. They have the ligand binds in a very center of these proteins. It's deeply buried in the hydrophobic part of the protein. As hormones are very hydrophobic molecules, but the ligand somehow has to go in there and get out of there so that it can activate and deactivate the protein function. Okay? So the questions that we would like to answer have been working on this for many years. The answers work actually. How the hormones enter the ligand binding pocket. How do they get out of there? So that's it, how they associate and how they dissociate. What are the molecular bases for high affinity, low affinity, or isotope selectivity? Can you bind better to this isoform and not to that isoform? So this is very important questions. We want to understand also molecular basis for nuclear receptors related diseases. It's very common, let's say you have some mutation. Rodrigo Speaks has one of these mutations. He's always joking about it. If you have one mutation, let's say I have a mutation that prevents me from my nuclear receptor working top-notch shape. So that might have introduced some metabolic diseases. Okay, just joking. All right. So we want to understand the impact of mutations and mobility and stability. There are hundreds of reported mutations that causes diseases. Okay? Usually they are very severe. Okay? So we want to understand nuclear receptors, biophysics. So just to make this a long story short, it's getting already late. There are the old model, still today, there's a very heated debate about how do ligands get in, how to get out, what is the structure. It's because of this picture that was put forth in 1998. It's like this. without the ligand, the protein, so if this is the structure with the ligand, without the ligand, this is structure, the crystal structure with the ligand. If you compare both the structures, they are very similar. The only difference is this helix 12 here. In this, without the ligand, the helix 12 is open, and with the ligand, the helix 12 folds here, as you see there. So it's called a mousetrap mechanism. Without ligand, the mousetrap is open. Ligand get in, you have to wait a fashion. The mousetrap closes. Okay. So this idea actually crystallized in people's mind. There are objections to this. Okay. We are one of the objectors to this picture. Not at least not like that. Not like a gate open and closed gate. So we have carried out several studies to understand this. And one of these, the very early studies that we studied, so if you want to understand how ligands get out of the enzyme, there is no hope just running a simulation, regular simulation, and forever, and trying to see a ligand release from its binding pocket, because the affinity is very high. So we have our hydrophobic molecule sitting in a very hydrophobic environment, and there's no reason for it to go out to the solution. So what Leandro did that time was to use a technique, very old technique actually, called enhanced sampling. Now there are many types of enhanced sampling, but it's locally enhanced sampling. It's like this. You have your ligand, you have your protein. What you do is you create many copies of your ligand inside the protein, replicated copies, but the one ligand does not interact with each other. So you can do this. You can just turn off interaction between the replicas of the ligand inside the protein. And the interactions between the ligand and the protein, you divide it by the number of copies. So that each copy sees only a fraction of the barriers due to the protein, but the protein sees a collection of ligands, each one with its interaction divided by a number n of copies, but you have n copies, so the protein sees a fuzzy ligand there. So with this, you are able actually to try to explore what are the multiple types of pathways that you can have. You have multiple copies, they are random velocities inside, and one will try to go there, the other one will try to go there. But at some point during protein fluctuation, it will find a way and escape. So that's what we have repeated these experiments many times, and then actually found three main types of pathways. One is the actual mousetrap, as this was one here. It's a helix 12, slightly moves aside and then ligand gets in. But there are other more interesting pathways here. Later, this was years ago, 10 years ago. Later, we tried to understand the likelihood of these paths. So we actually, once we have the path, we actually try to pull the ligand out so we applied an external force very smoothly, and we could compute the profile of this ligand binding, so using this steered molecular dynamics, so we can actually gain an idea what was the most probable escape pathway. It turns out that was not the mousetrap, it was something else, and we have a reason for that. We're still hoping for experimental confirmation on that. But I will skip a couple of slides on this one here. Okay. And just close with one of one of these results that we think it's interesting. We're going to try to go fast here. So TRIAC is a natural ligand for this thyroid receptor. Okay. It's a metabolite, and it's known experimentally that it binds better to the beta isoform. So now I'm back to this isoform selectivity problems. So this is what experimentally known. So in Igor's lab, they solved the structure for the TRIAC inside the protein for the TR alpha isoform and the TR beta isoform. So this is just part of the protein. So these are the key residues that are binding to your ligand. These are actual data. These are electronic densities, actually, this grid, blue grid that you see. If you look closely at this structure, you're going to see that the ligand is making many more interactions, hydrophilic, strong interactions, hydrogen bonds with the protein in the TR alpha as compared to the TR beta. The interactions here seems to be much stronger between the ligand and the protein in the TR alpha, not in the TR beta. But the binding affinity is higher in the TR beta. So there is a paradox, okay? So in terms of the crystal structure says the ligand should bind better TR alpha, but in terms of experimental data, the measured binding affinity is higher in the TR beta. So the paradox is that how that can happen. So this binds stronger to the protein. However, this paradox is just apparent because in addition to protein, you have water, you have ions in your environment. So in a simulation, you can compute all that. Those was very simple. You look at the TR alpha and the TR beta interactions. If you look at the interactions between the protein, the ligand and the protein, you see that of course it's higher. The interaction is higher, it's deeper, in the more negative with the TR alpha than with TR beta. But when you take the whole environment, then they are very nearly the same interaction energies. So this means that in terms of enthalpy, the binding enthalpy between the ligands and one isoform of the protein and the other isoform of the protein are very similar. So if there is any difference in binding affinity, which is a free energy quantity, that quantity should not come from enthalpic reasons, but maybe perhaps from entropic reasons. Okay. So we examined this, and we are able to actually show that there's entropic reasons for that. So here is just a cartoon, a superposed structure of the ligand inside the TR beta cavity, and here inside the TR alpha cavity and TR beta cavity. So you see that the ligand moves more freely inside the TR beta cavity. Actually, the TR beta cavity is able to hold a few molecules, water molecules, as opposed to the TR alpha cavity. So the ligand has more flexibility, more conformational freedom inside the TR beta cavity. So when you have flexibility or conformational freedom, means that you have higher entropy. Okay? So Leandro went there and devised a nice strategy to compute this entropy, which is something very difficult to compute usually from a simulation. But he was able to define a method to compute the entropy by looking at the actual conformations, comparing conformations with different energies and the same energy. So they actually, if you span the conformational space here given by two properties, in TR alpha you have this area for conformational sampling, and TR beta have a much larger area. So if you compute the ratio between these two areas and take the logarithm, you have the free energy difference. So this is what happens. So he computed this, the difference is very small, but it agrees with experimental estimates. Okay? And not only that, you were able to see who is behind this difference, who is behind these differences. And, okay, I can jump, I don't have to jump that. So what he did was, he identified key residues that were responsible for this behavior, this difference in behavior. And one in particular was a serine to asparagine residue in position 331. So what he did is that he took TR beta and made this mutation, and the behavior resembles TR alpha. He took TR alpha, made made this mutation in the computer, repeat the simulations, and then the behavior resembled TR beta. So he was actually proposing that if you do this mutation in one of the residues, the protein behaves as the other isoform. If you take the other protein and mutate to that guy, it reverses. So this is actually experiments. This was an asparagine and a serine. So if you take TR beta, this is TR beta, this is TR alpha. See the difference in affinity. The lower is higher affinity. So you take TR beta, mutate the residue, it behaves like TR alpha. You take TR alpha, mutate the residue, it behaves like TR beta. So this experimentally confirmed the activity. So this was very nice. Okay, and so my last slide before the economy is a very recent work that has been carried out by Melina, and it has been recently found that these nuclear receptors, not only this activity related to helix 12 motions and stuff like that, but these molecules are phosphorylated. I mean, you have a residue in a part of this protein, comes another enzyme, and binds a phosphor, transfers from the ATP, a phosphate group, into the protein. So it's called phosphorylation of the reactor. So when the protein, one of these proteins, get phosphorylated, it starts to express the wrong type of genes. So this is, for instance, something that happens to a ligand called PPARgamma, which is a target for anti-diabetic drugs. Several of these anti-diabetic drugs also induce obesity. Okay? And it has to do with this double action between controlling the enzyme, controlling the insulin sensitivity, but also the metabolism of lipids. So one of this finding is that if you design a ligand that does not activate very much this PPARgamma, but you actually prevents it from being phosphorylated, then you gain something. You gain insulin sensitivity activity without the deleterious effect of weight gain. So the idea is that this PPARgamma is phosphorylated by this cyclin-dependent reactor enzyme, and we wanted to understand what is the structural aspects of this. How is this CDK enzyme, how does it bind to this PPARgamma? So that was the idea. So there's no crystal structure for this, but there is a structure for a cousin of this CDK, it's a CDK2, bound to a peptide. Then this cyclin that CDK2 bound to a peptide, there's a crystal structure. So what we did was the following. We used this as a target, and we use a different type of enhanced sampling, which is called adaptive biasing method. In general terms is this: you have your protein, you have your target structure, you apply some sort of general forces on your system so that it brings this guy towards this configuration here. So it drives your system slowly and smoothly into the configuration, in the final configuration, the target configuration. So you don't know CDK5 and PPARgamma, how they are related, but you know CDK2 and a peptide. So you use the CK2 and peptide as your target structure, and you tell PPARgamma and CDK5, guys, you arrange yourselves until you get your structure that is similar, at least this part of the structure that is similar to the target. So this is called ABF, and we do this, and in the end you have very good matching between the target and the initial structures. But this work was carried out by Melina, and so Melina were able to propose 4 different types of models for the association between CDK5 and PPARgamma. So she started doing simulations on these models to see which of these models is actually stable. So then we start to understand how stable or how bad, how good these models are. And in the end, one of these models actually turns out to be better than the other models. And we also investigate the implications of different ligands into this. Once you have this model, you can start to understand what if I have different ligands bound to my PPARgamma that will prevent it from being phosphorylated by the CDK5 enzyme. So that's the idea, and so she was able to at least provide some clues of what is happening. She also, of course, identified very important regions where CDK5 and PPARgamma binds to each other. So regions that are important for the binding between the two proteins. Okay? So in the end, it's my simple conclusions, one simple phrase, is that I hope have shown to you that our modeling can be worked in very close partnerships with structural and functional molecular biologists, and we are able, hope, to provide some molecular level interpretations that are very difficult to find from experiments alone. Okay, so this is the synergistic action that molecular dynamics and other types of proteins are good for. So, with that, I close and I thank you. Here's a list of people that has contributed, with whom we work very closely. Thank you all.
Douglas, one of the results you mentioned that the error bar for Igor, I guess, it's very large.
Error bar?
For the
What's it this?
1.5, something like this. For the Igor.
Protein? Oh, resolution? Oh, resolution is very high. Very good. 1.7 angstrom resolution.
But there's still a lot of room for, so how can you really distinguish between the conformations?
No, let me see if I understand. No, I'll go back and say stop there. This one? Which work? I don't understand which work. Not this one, not this one, not this one, not this one, this one? No.
Next one.
Next, above? No.
Okay, doesn't matter.
Doesn't matter. I don't know which structure you're talking about.
You try to use the model to distinguish between conformation and the biological implication, right?
Mm-hmm.
But the difference in the conformation for the simulation seems smaller than the experimental error bar, so you could see. No.
In the simulations, the structure fluctuates a lot more than the reported. Let's say the crystal structure does not report the fluctuations. You have something called the beta factor that gives you some sort of the beta Waller factor in solid state that gives you the idea of the average position, but you don't see, there are no error bars in the structure. Say, oh, this is the resolution can tell you a little bit. So if it's 1.7 angstroms, for instance, resolution, this means that you have the certainty of finding a position for a certain atom, so heavy atoms, it's 1.5 here or there. So grossly speaking. So this type of resolution is very high. So usually 2.5 is a so-so, 2.5 angstroms. Because then you can even miss a hydrogen bond, for instance. but 1.7 is. So the fluctuations in our structures are way higher than what you would expect from the tissue structure alone.
Any more questions?
So just so that I learned something. The difference you determine by visual inspection of the models, is that so?
So,
You determine the difference by visual inspection. You know,
Yeah.
go to the any of them.
yeah, no. Mhm.
Go to the more advanced one, the one that's turning alpha and beta.
Yes,
models.
okay. Okay, I understand.
So this, how do you determine the difference between the two structures, other than by taking a visual look at it?
Oh yeah, yes. The crystallar, these are crystal structures. Okay, they obtain the two structures, okay, and then they compare,
Okay.
and then they look at the contents. And say, well, this is closer to that, this is closer to this is making hydrogen bond here. So they cannot compute interactions unless they put some sort of potential between the particles. So this is just binding. When you look at the binding, you say, "Hey, this looks that it binds much better this isoform," but they don't. They cannot quantify how the binding is. Okay, is that what you're saying? Not really.
Ask
More?
again later.
Okay, now I understand. So now we compute, and the interactions is actually higher, more negative, let's say, with one of the other. So that the simulation agrees with. So this, when they claim that this makes a stronger binding to the protein, yes, we agree. If you compute the interaction between the protein, the ligand and the protein, it's actually more negative for this isoform. So this is correct. It's further? Okay.
No question? Thanks. Let's thank again Munir.
Thank you.